Hello, my name is Chris Solomon, and I'm here to deliver a lecture about bite technology. I wanted to illustrate the lecture about bite technology using the only compound which I have an experience with, which is blinatumomab. But unfortunately, to understand blinatumomab, you also need to understand acute lymphoblastic leukemia. So, in this lecture, we are going to have a whistle-stop tour about acute lymphoblastic leukemia. We're also going to we're going to discuss, going to illustrate the pharmacology of bite technology in general using blinatumomab. We're going to discuss some of the unique adverse effects that you get with some of the T cell directed therapy in general, but particularly with blinatumomab. Blinatumomab is a small protein, and this has resulted in a few interesting pharmaceutical particulars, which I will discuss. They have led to some issues within our trust. So I also wanted to cover the delivery of blinatumomab and most likely future bite drugs to patients at the Royal Marsden Hospital. So to really understand bite technology and blinatumomab in this particular case, you really need to understand acute lymphoblastic leukemia. Now, it's beyond the scope of this lecture to really discuss acute lymphoblastic leukemia in great detail, but I'm going to cover in the next couple of slides. I will cover a little bit about acute lymphoblastic leukemia. This is a peripheral blood film pinched from Cancer Research UK, and these circulating, stained cells here are blasts. You can tell they're blasts because normal peripheral blood contains cells which have a large component of cytoplasm. The blast cells have an absence of cytoplasm, and this is characteristic of a leukemia. Blast cells which have come out of the bone marrow and entered into the periphery. Epidemiologically speaking, acute lymphoblastic leukemia is a very rare disease, accounting for between 700 and 800 cases per year, usually about 750. Most cases are diagnosed in patients who are less than 10. 53% cases are diagnosed in patients less than 10. The prognosis is dramatically different between the younger age group and the older age group, with the younger age group achieving far better remission rates, cure rates, than the older group. In fact, there is some discussion to say that whether they're almost two different types of diseases. Patients who are diagnosed as children, or pediatric patients with acute lymphoblastic leukemia, have the potential, significantly quite high potential, to retain a cure with chemotherapy alone. However, patients in the older age group have a much poorer prognosis and are unlikely to be able to obtain a cure with chemotherapy alone. This patient group will almost definitely have to have the only option for a long-term cure or disease control is through a process known as stem cell transplantation. Broadly speaking, there are two subtypes of acute lymphoblastic leukemia: B cell and T cell, although there is a biphenotypic subgroup which have facets of both B cell and T cell ALL. To understand acute lymphoblastic leukemia, or leukemia in general, I've pinched this slide from Wikipedia. Which is an overview of hematopoiesis. At the top, we have the hematopoietic stem cells, which are the stem cells which produce all of the components within your blood. Broadly speaking, on the left-hand side, we have the components of the innate part of your immune system, and on the right-hand side, we have the components of the learned immune system. Essentially, these are the phagocytotic components of your immune system, your mast cells, which are the cells responsible for producing histamine, your red blood cells, the cells responsible for carrying oxygen in your blood, and your thrombocytes or platelets cells are involved in coagulation. Your T and B cells and your natural killer cells form components of your learned immune system, i.e., this part of your immune system recognizes previously exposed fungi, bacteria, viruses, and starts an immune response against them. Of note, the plasma cell at the bottom here is the cell responsible for producing most of the antibodies produced in the human body, and the natural killer T cell, which we'll discuss in the next few slides. So it was beyond the scope of the lecture to really discuss ALL treatment in children, and probably beyond my real scope of practice. So I did a very brief overview of acute lymphoblastic leukemia in adults, and the gold standard for acute lymphoblastic leukemia is the UCAL14 trial. And so essentially, if you're over the age of about 25 and are diagnosed with acute lymphoblastic leukemia in the UK, you'll be put onto or be offered the UCAL14 trial. And even if you are not put onto the trial, the chances are you'll be following the trial protocol, whether you're a P or T cell ALL patient. Now, unlike the pediatric protocol, which is rather similar to the UCAL14 protocol, the UCAL14 protocol is essentially a protocol which is just used to put a patient into remission prior to a stem cell transplantation. And it's only a stem cell transplantation which offers patient a long-term cure or disease control. And most patients are able to attain a remission either in the first arm or the second induction arm. But this does raise a few questions. So what happens if a patient never achieves a remission in UCAL14 or relapses before we can give a stem cell transplantation? There is always going to be a time allowance so that we can actually deliver a stem cell transplantation. Patients do, unfortunately, sometimes relapse from that period of completing their chemotherapy within UCAL14 to them actually receiving a stem cell transplantation. Now, traditionally, we have offered a salvage regime with combination chemotherapy, and our salvage regime is FLAG-IDA. FLAG-IDA is combination chemotherapy involving a mixture of 3 chemotherapy drugs and a growth factor. Interestingly, kind of uniquely in oncology general, we try to exploit GCSF's ability to put cells into the stem cell cycle. And in theory, that may make them more vulnerable to some of the effects of the chemotherapy. So actually, on the first day of this chemotherapy regime is actually GCSF, not chemotherapy. The backbone of this, or the keystone of this protocol, is really cytarabine. Fludarabine added in, although it's a potent anti-neoplastic drug itself, it's actually used primarily because of its interacting qualities with cytarabine, pushing a kinetic interaction which sort of prevents the metabolism of the cytarabine, therefore potentiating the effects of the drug. And the drug and cytarabine is really the drug which is sort of going to do the business in this protocol. The idarubicin is relatively mild in comparison to the other anthracyclines, but it has a broad spectrum of activity and is relatively non-toxic. FLAG-IDA response rates are actually relatively good. FLAG-IDA response rate actually relatively good. Remission rates are between 40 and 50%. However, still 50% of patients will have residual disease and would be unable to have a stem cell transplantation.